Hello students, welcome to EPG Pathshala. My name is Dr. Priyanka and I teach Anthropology at the Department of Anthropology, Dr. Hari Singh Gaur University, Sagar, MP. Today, we are going to discuss about the module on case studies of natural selection in humans. This module comes under the paper of Human Population Genetics. So let's see that what you should expect from this module. In this module, we will be showing you the illustrative example of several types of selection, particularly those that show evidence of rapid evolutionary change in recent human evolution. By the end of this module, you will be able to understand how natural selection works at the population level. Before we start giving you the case study, let's understand about the process of natural selection. The theory of natural selection was given by Charles Darwin. His idea of natural selection was inspired from the idea of Lamarck, Lyell, Malthus, Wallace, and many other scholars. In the year 1859, Darwin published his book known as Origin of Species, in which he gave his idea of natural selection. The basic idea or the principle behind natural selection are: number one, biological variation. Biological variation is a phenomena which is present in each and every species. Secondly, Darwin was influenced by the idea of Malthus, which says that the species are capable of producing offspring at the faster rate than the food supply. That means there is a constant struggle for the survival because of the high competition for the resources. Some of the individuals in a population have favorable traits which increases their chance of survival and reproduction. You should remember here that favorability of a trait is determined in the environmental context. The traits which are favorable, they make that particular organism to be better adapted or suitable in the given environmental condition. And then these traits are passed on to the next generation. Therefore, we can say that the traits are transmitted. Because individuals who have favorable traits, they contribute more offspring to the next generation than those who do not. Individuals who produce more offspring in comparison to others are said to have greater reproductive success or fitness. Reproductive success is the individual's contribution to the next generation. It is basically the number of the offspring an individual produce and rear up to the reproductive child. So more the reproductive success of the individual, the more fit it is considered. Over the long period of geological time, successful variation accumulates in a population, so that eventually later generation may be distinct from the ancestral ones. Thus, in time, a new species may appear. Darwin also realized the importance of geographical isolation. Geographical isolation contributes to the formation of a new species. But how? Let's see. As population of a species become geographically isolated from one another for whatever be the reason, maybe due to some water body or maybe due to some mountain, they begin to adapt to different environments. And over a period of time, as population, they became different due to responding to the different selective pressure. In terms of genetics, evolution means any genetic change in the population of a species which makes it distinct. Several factors can alter the gene pool of the population, and natural selection is one such factor that contributes to making the gene pool more distinct. Selection means selection of favorable traits by nature. Every trait is determined by genotype, so selection in the non-random differential retaining of favored genotypes. any genotype is more fit, is favored and propagated, such that there is a change in the allele frequency over time. The principle underlying natural selection is that any gene which confers a disadvantage to its carrier within a given environment will decrease that organism's chance of survival to reproductive age and thus decrease the frequency of that gene in the gene pool. Let's understand with the example. Suppose there are two populations A and B carrying two different kind of a gene A and B. Individuals carrying gene A are more successful in reproduction than individuals carrying B. Then frequency of A gene will tend to increase generation after generation at the expense of gene B. Any trait which gives an organism a better chance at survival at a given environment will increase the organism's ability to grow and reproduce, and it will increase the proportion of gene for that trait in the gene pool. Selection indirectly alters the gene frequency by acting on its carrier by increasing or decreasing their ability to reproduce. Through natural selection shaped the human population over time, the rapid change are not only as a result of natural selection, but are instead changes in the environmental condition. A trait or its corresponding genotype may be more fit in a given environment and may not be fit when the environment setup changes. Study based on human genetic and morphological variation have allowed us to understand a number of significant examples of natural selection in today's human evolution. Let's view and understand them one by one, and let's start with the first example of sickle cell anemia. The story of natural selection and hemoglobin molecule is a classical example in anthropology. It is an excellent example of balancing selection, rapid genetic change, and the effect of cultural and ecological influences on selection. Hemoglobin is a protein of the blood that transports oxygen to tissues throughout the body. Normal hemoglobin molecule consists of 4 protein chains, composed of 2 identical chains of alpha and 2 identical chains of beta. The alpha chain are 141 amino acid in length, and the gene for this chain is present on chromosome number 16. The beta chain, on the other hand, is 146 amino acid in length, and the gene for this chain is present on chromosome number 11. The normal form of beta hemoglobin gene is known as the A allele, and the people with AA genotype have hemoglobin which functions normally in transferring oxygen. SS genotype result in the manifestation of a disease called as sickle cell anemia. In this condition, single mutation replaces the 6th amino acid of beta chain glutamic acid with the amino acid named as valine. The disease can cause the red blood cell to become distorted and changes the level of oxygen, and the donut shape turn to the shape of the sickle. Hence, the name sickle cell has been given. The deformed blood cells do not function properly and do not carry oxygen effectively. This causes serious problem throughout the body's tissue and organ, and typically leading to death before adulthood without substantial medical intervention. With respect to natural selection, relative fitness is assigned to different genotypes. In this situation, we have 3 different genotypes: capital A, AS, and SS. The genotype AA is reported to have highest fitness. Genotype AS slightly lower relative fitness, whereas the genotype SS is expected to have the lowest fitness. The main focus is laid on A and S allele. Because S allele is harmful in homozygous condition and a selection against allele is done. Allele S is mutant and has the lowest frequency. Data from many human population certainly fit this prediction. For example, the allele frequency in a number of population throughout the world are A being equal to 1 and S being equal to 0. In some other cases, S is not 0 but is relatively low. Such frequencies are consistent with the model of selection against an allele and would simply be a good example of such selection if not for the fact that the number of human populations do not fit a general model of natural selection. On the contrary, some populations in the world have higher frequency of S allele. These higher frequencies than expected under the model of selection against the S allele poses a problem. How could the frequency of a harmful allele be higher than expected? There are two possible answers to this situation. One is the drift, that means by chance the frequency of harmful alleles have been drifted upward. The other explanation is balancing selection when there is the heterozygotes, as we see in this situation, which confers higher fitness than some of the two other genotypes. Balancing selection. Balancing selection refers to a selective process by which multiple alleles get added and are maintained in the gene pool of a population. A moderate to higher frequency of S allele are found in the populations in a part of West Africa and South Africa, as well as in a part of Middle East and India. Populations that have history of malarial epidemics tend to have higher frequency of S allele and can be best explained by balancing selection. Presence of an S allele renders the red cell inhospitable to the malarial parasite, thus protecting the individual from its effect. In a malarial environment, the heterozygous AS has the highest fitness, followed by AA genotype, and the SS genotype still have the lowest fitness. Sickle cell hemoglobin also provide a classical example of how culture affects genetic evolution in a human population through the process of natural selection. The frequency of hemoglobin S increases in a part of Africa because of practicing agriculture. In this scenario, forests were cut and planting and shifting cultivation began. This changes the soil chemistry and causes the environment to be more warm and sunlit. These conditions are appropriate conditions for the mosquitoes and mosquito parasite to live. As a result, malaria increased. Those with the heterozygous genotype were found to have the highest fitness as they were resistant to malaria as well as do not suffer from sickle cell anemia. So, frequency of sickle allele increases, but this also results in the increase of the homozygous genotype for sickle cell allele. Individuals, however, they were selected against by nature due to the health implications of homozygous individuals. The figure shows the distribution of Duffy alleles all over the world. As we can see in this picture, the highest frequency of Duffy negative allele is present in the region of Africa and surrounding Africa. Next case study to show the operation of natural selection in nature is Duffy blood group and malaria. As we know, a number of genetic traits show evidence of natural selection because of malaria, including hemoglobin alleles, thalassemia, enzyme deficiency, G6PD. Duffy blood group is also one such trait. Duffy blood group is defined by the presence of antigen on the surface of red blood cells. The gene of Duffy blood group is present on chromosome number 1. It consists of 3 codominant alleles, FA, B, and F negative. FA allele code for antigen A, FB allele code for antigen B, and F negative allele code for antigen neither A nor B. Individuals who are homozygous for Duffy negative allele cannot be infected by Plasmodium vivax, which causes vivax malaria. A selection of Duffy negative allele are expected to be there in population experiencing vivax malaria, ultimately leading to fixation of this allele. The highest frequencies of Duffy negative allele is found in Central and South Africa, reaching a value of 1, which means 100% in numbers of populations. Moderate to high frequency is found in Northern Africa and the Middle East, at low frequency in a part of India, and or close to zero in native population throughout the rest of the world. Supports for natural selection for Duffy negative allele come from the molecular studies that have examined DNA sequence at and near the location of Duffy blood group gene. The evidence show for the Duffy blood group and neighboring DNA sequence have reduced in variation. The reduced variation for those with the Duffy negative allele confirm that selection has taken place. However, does this mean that the selection initially resulted from adaptation to vivax malaria? The answer is not necessarily. Livingstone in 1984 present another hypothesis that can also explain the negative correlation between Duffy negative allele frequency and prevalence of vivax malaria. He suggests that there are already a high frequency of a Duffy negative allele in some other diseases. Consequently, these populations were already resistant to vivax malaria when it spread into Africa. But because almost everyone was immune, the disease never took hold. This alternative continues to be a possibility, although estimates of the date of initial Duffy negative mutation from coalescent analysis suggest that it arose fairly recently, at about the same time as the origin of agriculture and the spread of malaria. If so, then vivax malaria was responsible for fixation of Duffy negative allele in Africa. Another example which we are going to discuss is CCR5 Delta 32 gene mutation. On the microevolutionary level, we can see the same thing when looking at the variation of CCR5 Delta 32 mutation as Duffy. This mutation causes the deletion that results in the absence of CCR5 receptor on the cell surface. This condition do not allow the pathogen to enter in the cell and thus infect the cell. This description is clearly evident in the given picture, where the absence of CCR5 receptor do not allow the pathogen to enter inside the cell. The CCR5 gene is located on chromosome number 3. This gene is responsible for the CCR5 protein, which functions in resistance to certain infectious diseases. A mutation of CCR5 gene results in the deletion of 32 base pair section of DNA sequence of CCR5 gene and known as CCR5 Delta 32 mutation. In European population, Delta 32 mutation has a frequency of about 0 to 14%, but it is absent in rest of the world. Statistical analysis of DNA sequence near this locus provides strong support that the distribution of this allele has been shaped by natural selection. Heterozygotes with one CCR5 Delta 32 allele show partial resistance to HIV, and homozygotes show almost complete resistance to AIDS. However, AIDS has been known only in a short time in human history, and therefore it could not have been responsible for the initial elevation of this mutant allele in some of the European populations. Most likely, higher frequency of CCR5 Delta 32 arose because of the selection related to some other disease such as bubonic plague or smallpox in European history. Human skin color, also known as pigmentation, is quantitative trait that shows an immense amount of variation between human groups around the world. We see people of a very dark skin color to the people of extremely light skin color. Skin color tends to be darker at or near equator and lighter as it moves away from the equator. Skin color, UV levels, and distance from the equator are highly correlated parameters. Darker skin color act as a protective barrier against UV radiation, which is highest at or near equator regions. Therefore, this trait is selected against light skin color in this region. In this figure, we can see that the skin color tend to be darker at or near the equator and decreasing with the increasing distance from the equator. UV radiation is stronger at the equator and diminishes with increasing distance. Skin color level of UV radiation and distance from the equator are all highly correlated factors. Pigmented skin act as a protective barrier against UV radiation. Therefore, dark skin are preferable in the areas at or near the equator. Let's see how natural selection helps in the adaptation to high altitude. A change in the adaptability was observed among the humans as our ancestor expanded out of Africa and spread across the world. One particular challenge occurred in adapting themselves to the high altitude and overcome the physiological stress. Some populations live as high as 500-4,000 meters above the sea level. When a low native enters a high altitude environment, the hypoxic condition can be encountered by various individuals and an adaptive response is further generated, such as increase in the red blood cell and increased respiration, increased chest dimension, greater lung volume relative to the high altitude. Some adaptation occurs in infants and children who move to high altitude through the process known as developmental acclimatization, where there are changes in the body during the growth process when adapting to an environmental stress. Those who move to high altitude show an increase in aerobic capacity compared with those who stay at the low altitude. Further, the younger the children who moves to high altitude, the greater is the chance of their being adapted to the high altitude. There are some genetic influences on high altitude environment, then some physiological and biological trait as a result of natural selection. As can be seen from the genetic makeup of the respiratory component of the Tibetan and Indian population are significantly different. Indian population is a native inhabitants of Indian Plateau in South America who descended from human population moving into the region about 11,000 years ago. The other group, that is Tibetan population, is the native inhabitants of the Tibetan Plateau who colonized the area about 25,000 years ago. These two populations differ in the level of oxygen in the blood and the level of oxygen saturation in the blood and the level of hemoglobin concentration. The important lesson here is that there may be different paths of selection that operate differently depending on the type of genetic variation available, which in turn is influenced by variation in mutation and genetic drift. Besides multiple adaptive solutions, genetic studies of high-altitude adaptation provide another example of rapid pace of recent human evolution because the high-altitude population have been in those environments only within a recent evolutionary time frame. Now let us consider a different sort of selection, evidence of adaptation to changing diet. Human infants are nourished through breastfeeding. Infant mammals produce the enzyme known as lactase, which allow lactose, which is a milk sugar, to be broken down and digested. The typical pattern in mammal is to shut down the lactase production after the infant is weaned. After this, the mammal can no longer easily digest lactose. Today, many humans have developed lactose intolerance, that they cannot produce the lactase about after 5 years of age. The physical effect of lactose intolerance can vary and include bloating, cramps, distension, and acute diarrhea. The interesting fact is that although many populations are lactose intolerant, while others have no trouble in digesting lactose as they continue producing lactase enzyme throughout their lifetimes. The gene controlling lactose activity is found to be present on chromosome number 2. There are two lactase activity alleles, LCTR and LCTP. The R stands for lactase restriction, that codes for shutoff of lactase production after weaning, whereas the P stands for persistent production of lactase. Apart from some minor mutations that are found in low frequency, LCTP allele is dominant, so individual with one persistent allele, either LCTP LCTR or two persistent allele, that is homozygous LCTP, can more easily digest lactose. The recessive homozygote, that is LCTR are lactose intolerant. Lactase persistence has an interesting geographical distribution. It is found highest in Northern Europe and moderate in Southern Europe and Middle East. It is found very low in African and Asian population on average, although some African population have moderate to high frequencies. Variation in Africa is particularly revealing because some populations are found with high frequency of lactase persistence and some have the very low levels of lactase. The critical factor that explains global variation in lactose persistence is diet. Population indulged in dairy farming tend to have higher frequency of lactase persistence allele. The fact can very well be explained by natural selection, where lactase persistence was selected for the population engaged in dairy farming because of the nutritional advantage among individuals who are able to digest the milk. A European cattle study revealed that the geographic distribution of various protein genes was correlated with the levels of lactase persistence in humans and the location of prehistoric sites associated with the early adoption of cattle farming. As humans adapted to a diet including milk, they also selected cattle that provide better quality and quantity of milk. The estimated age of lactose persistent fits the archaeological evidence for the origin of the age of domestication of cattle. Cattle farming began in North Africa and the Middle East around 7,500 and 9,000 years ago, which fits with the estimated age of 8,000 to 9,000 years for the T mutation in European. So students, let us summarize this module. In this module, we have seen a number of significant examples of natural selection. Hemoglobin S we saw as an example of balancing selection. The next in the row that we discussed was a fixation of a Duffy negative allele. Duffy negative allele fixation is a genetic adaptation related to the spread of Vivax malaria. Next, we saw the case of CCR5 Delta 32 gene mutation. The association of Delta 32 mutation of the CCR5 Delta 32 gene with the resistance of HIV and AIDS is highly interesting. However, the evolutionary history of this allele suggests an initial adaptation to a different disease other than HIV/AIDS, which perhaps can be smallpox or bubonic plague. A striking example of environmental adaptation is a UV radiation and the correlation of this with the skin color. Another genetic change reflects the propensity of our ancestor to adapt genetically at a high altitude environment to the physiological stress of the lowered amount of oxygen, although in a different ways. Thank you.